mode, you should see it properly.
Perfect.
Okay,
Perfect.
fantastic.
Perfect.
So,
Yes. Away
we
you
did
go,
it
Eduardo.
faster. Yeah, so that's great. Sorry about that. So, as I said, I'm Edouardo Puglisi. I'm a professor of agricultural microbiology. I'm based in Italy, Piacenza, Milan, and I'm coming from agriculture, food, environmental science. And I'm mainly a soil microbiologist, but I've been also working quite a lot on pollutants, and as we're going to see today, also in other microbiomes. And we've been working on the plastisphere, I would say, from the last 12, 15 years. And of course, many of the activities we carry out in my group are also related to the MINAGRIS project, where we are doing quite a lot of work. So I will also present in some evidence from MINAGRIS, but also other evidence from our work and, of course, other literature data. So the topic of today indeed is the plastisphere and how this is connected to soil, plant, animal, and human health. As a starting point, of course, we can just introduce plastic. Most of the people here who come, of course, are already familiar with that. So we're talking about polymers of different shapes and different characteristics. The plastic production, which is, let's say, a characteristics of the contemporary era in the last century, is steady increasing. So this is some of the data you can see about the current data and the current trends. And we see, despite some of the problems we will also highlight today, steady increase in the use of plastic. And we can also see that the fate is changing a little bit because we got different fate, different uses that goes from recycling, discarding, incinerating, and so on. So there is, of course, a strong complexity related to this. Talking about plastic, we also need a little bit a little bit of definitions. And of course, here we got plastic and bioplastic as well. This is a classical scheme that we've seen. And we can, from European Bioplastic Association, we tend to distinguish depending on the sources. So you may have fossil-based or bio-based plastics, and depending on the fate. And here on the fate, of course, we got the topic, the issue of biodegradability, which is, of course, extremely important. And a normal assessment is that indeed fossil-based plastics are mostly non-biodegradable. Bioplastic, they can be, but this is depending from a lot of factors. And both of them are growing. We should also state that there is, unfortunately, a confusion in the terms because nowadays the bioplastic defines both the, let's say, the one that could degrade, but also the one that are bio-based. So they derive from renewable sources, but for example, you can have bio-based polyethylene, which means that is polyethylene in any characteristic, it's just the source is different. And I have to say also about this that we should also always remember that it's not the origin, but it's the chemical structure that makes the characteristics and also the potential ecological impact of a molecule. And in this sense, of course, there is no difference between the two. One of the big issues with plastic, and actually this is, I think, in the last 20 years, one of the most important issues that we realized that plastics tend to fragment, okay? We can remember that a great scientist and a colleague, Professor Richard Thompson from Plymouth University, 21 years ago, coined the term in microplastic. He was observing the shores, he's a marine biologist, and he was observing these tiny, tiny particles. And what we are learning is that these are increasingly present in the environment. As you can see, this is a recent paper that is showing us how the plastic can fragment to micro and nanoplastic. And this is, of course, making a lot of changes because we got a higher surface volume, which usually creates more problem. And this is, of course, related when we're going to talk about the plastisphere. And the point that indeed this material, they do not disappear. Unfortunately, they tend to disaggregate, and the disaggregation creates particles that tend to be more biologically active. So the vision of decades ago of the plastic of something totally inert, unfortunately, I would say for us and for the environment, has been changing. I'm an agriculture scientist. Minagris is a project that has been introduced before that is focusing on soil. And what we know is that around 10 years ago, and here I have to acknowledge a great colleague and a friend who is Luca Nizzetto, that is also the coordinator of a sister project, which is called Papillon, published on Nature a kind of call to action saying that the amount of microplastic in soil indeed was huge and was probably higher than the one that we got in the oceans. So it's a kind of new emergency. And And now we are learning more and more, as you can see in this scheme, about what is happening to the plastic once they reach the soil and agriculture system. Luke as well made, with some other colleagues, a very important document for FAO last year. I also have to remind that 3 weeks ago we had a workshop at FAO dealing with plastic in agriculture, and also people from MINAGRIS and the project were involved. And there is an important commitment. This report that you can see, all my data, of course, all my slides at the citation provided, is providing a lot of data. So for example, this graph we've seen here is showing us that these are the amount of plastics, so expressed as a percentage. The scale is 10 to the minus 4, 10 to the minus 3, minus 2. So we are talking about 0.1, 0.01%. But as you can see, these are data from controlled soil, soil where there is mulching film, which is a source of plastic, source from greenhouse, fertilizer, and so on. In all of them, there is microplastics, okay? Of course, with a higher level where certain use of plastic in agriculture are being made. And in the same report, there is a literature review. As you can see, 189 studies assessed dealing with plastic. And as you can see, most of them, the largest part of them, reported effects. And in these effects were already found at concentration that are representative in soil. So the main issue also for agriculture is that plastic are everywhere, and they are also already reporting some problems, okay? And this is a kind of call of action that in the last years has been raised. In this sense, I found particularly interesting this paper that was published a few months ago. Okay, we are in PNAS, so we are one of the best journals in the world. And here they made a meta-analysis, as you can see, of data set of more than 3,000 records about the impact of plastic on the plant physiology and the photosynthesis and chlorophyll content. And the data, as you can see from the estimations, are solid in showing a significant decrease in photosynthesis. So there is also effect on the plants. And as you can see on the left side, we can see that for a number of crops, okay, these are models. So we should take them into account as models. But as you can see, it's already possible to estimate losses of agriculture production in rice, in wheat, in maize, and so on, all around the world due to the microplastic pollution at the moment and for the future trends. So let's move to the topic of today, the soil plastisphere. So I'm a microbiologist, and the plastisphere is a microbial concept because it was defined for the marine environment by Eric Zettler a few years ago. And he called it basically a community of microbes that is growing as a thin layer of life, which is a biofilm. Biofilm are very well known in microbiology, on the outside of the plastic. So what they were stating is that the so-called plastisphere, so these microbial communities, is similar to the layer of the life on the outside of the planet that we call the biosphere. So basically they define a kind of new ecosystem. And we are learning that this ecosystem is quite different from the other ecosystem, including soil. So afterward, you can see this paper from Matthias Rehlig and other colleagues, was called the soil plastisphere. So basically the concept of plastisphere was then applied to the soil. And as you can see, there are a number of processes where in the soil environment, all the plastic particles we are having are interacting mainly with the microbes. And this is also affecting interaction with pollutants, with soil processes, and so on. So we are learning this concept. I will also now introduce some of our work. And actually, this is also a little bit of personal history because this one was one of the first studies we carried out. Actually, we were already studying plastic because, for example, we isolated some plastic degrading microbes from some glaciers with colleagues in Piacenza. But then in 2015, which is 10 years ago, so time flies, and we went to a landfill close to a university because some environmental agency indicated this landfill. And actually, as you can see from the picture, this was an agriculture field, okay? Quite astonishing because you can see the amount of plastics that were there. And this place used to be a landfill, as you can see from 1972 and 82, that received quite a lot of thousands of tons of wastes. Then the landfill was closed, and what was happening is basically that the pollutants that could either degrade or move were not there, but the other ones, including metal, glass, and so on, but especially plastic was still there. So we went there and we retrieved plastic films, as you can see, from different colors, because this was our way of classifying. We went to the lab and started studying it. So here you can see some electron microscope pictures of different plastic. Here you can see the green one and the red one as an example, before and after washing the biofilm. So what you can see is really the bacteria, the fungi, the archaea, all the microorganisms growing there. And the fact that when you wash it away, you can see the surface of the plastic, for example, this red one that is quite interesting with some holes and so on. And we collaborated with Polytechnic University, Professor Fracchi, who is also a partner of Minagris, assessing the level of degradation. And we found that all of them were polyethylene. The difference was in the additives. And there was also some little difference of degradation, because you can consider that at the time of sampling, the plastic were there for 30 years, okay? So, for example, the red one, you can see from these shapes, there was more evidence of degradation. When we did the microbiome, we could see that as compared to the soil and another controlled soil, the plastisphere had a completely different microbial community. And as you can see from the color, each kind of plastic had a different community from the others, okay? So each kind of plastic is hosting a different microbial community, a different plastisphere that is different, of course, from the soil. And looking at the kind of microbes, you can see many differences. For example, there is this increase of bacilli, which could be also interesting for application afterwards. On the right side, there is an index of diversity, and it's something interesting we found here, because this red plastic, as you have seen before, was the one mostly degraded. And what is happening is that its microbial community, as you can see from this graph, is getting a little bit more similar to the soil. And the same we see it in terms of diversity. Of course, there is a huge reduction in diversity as compared to the soil in the plastisphere, because it's a very specialized environment. But it seems that the most degraded one, the red, is getting a little bit closer to the soil. So it's a kind of probably evolutionary process that is taking a lot to bring it down. We went forward, so this is another study that arose from that, and this was in collaboration from colleagues of Turin University. Federica Spina was the first author, and Cristina Varese was also a co-author, where we isolated a number of fungi from the plastisphere, and we found that some of them had a certain ability of degrading polyethylene. Okay, so here you see the collection of strains of fungi, and then here you see the level of degradation. Main message here in both cases is that we are still dealing with polyethylene. As we know, polyethylene is not degradable. It means that the microbes are able to make some kind of attack, a little bit of reduction, but not a complete degradation, not at all. The only plastic for which this was found was PET for PETase. My guess is that probably microbes haven't developed yet the pathways to do it, but hopefully they will do it, because there is a continuous evolution. Going to Minagris, some details. In Minagris, among the many things we are doing, we are studying a lot of case studies. So around Europe, as you can see, there are very different soils where plastic is being used, and a lot of analysis have been done. In particular, in Piacenza, what we did in Cattolica was to study the plastisphere. So we took a sample of what we call mesoplastic. It means they are not micro, they're a little bit bigger, also because if you want just to retrieve the microbes, you need this. And we analyzed the microbial composition. This is just a glance of the results. The main point is that we found a strong effect of the different climate. So this is the distribution of genera around the microplastics, and also depending on the size. And again, we see the enrichment of certain groups of microorganisms. These kind of studies are going on. So here I'm showing you a collaboration with Professor Stefania Federici from Università of Brescia. She's also the coordinator of the cost action priority on plastic. And here we did an experiment, very recent, so it's not published, it's under review actually, where we tested different kind of plastics, and here we applied a whole genome sequencing. It means that now we extract the DNA and sequence all of it. And what we are finding is an enrichment of genes of interest. So the message here is that the plastisphere is selecting for microbes that are able to thrive on it. And maybe, hopefully, I would say, from this studying of plastisphere, we can also obtain biotechnological solution to try to tackle the plastic issues. I will move now to another topic, which is extremely relevant when we're talking about the plastisphere, which is the antimicrobial resistant threat. Okay, so here we are dealing, of course, again with a microbiological issue, but it's a medical microbiological issue. And this is a recent paper. I think it's Science, whatever. Anyway, it's published in 24. And this is an estimation of the number of deaths around the world that can be attributable to antimicrobial resistance. And these are the difference between 1990 and 2021. Okay, the number are quite huge because we are talking about thousands and thousands of deaths, and there is a change of the trend. So the simple message here is that antimicrobial resistance is one of the things that I would say as humanity we need to take care most of because lot of people are dying from it. So what is the relationship with plastic? There are now a lot of studies. This was one of the first published on this journal, which is quite interesting because here they assessed a controlled soil, a soil treated with manure, okay, and they compared the number of antibiotic resistant genes and the proportion of pathogens in the plastic treatments as compared to the control, both in the soil, both in the manure. And manure is relevant because, of course, in manure we got a lot of antibiotic resistant genes. In the manure and in many organic biomasses we got plastics. And this is a combination that could be of concern because this data and other data are showing us that when you got plastic, the proportion of pathogens is increasing and the proportion of antibiotic resistant genes is increasing as well. This is a table, not easy to read because a lot of data, but of course this is from a review just published this year. And here you can see a number of microplastics. So on the first column, the type, polystyrene, polyethylene, polypropylene, and so on. The potential, the size, the concentration level, and the genes, antibiotic resistant genes, TET8, TET-G, TET-X, and so on, and the changes of ARG abundance. So all of this data, this is a review, this is a meta-analysis, are showing us that there is an increase of antimicrobial resistant presence and potentially transmission. We have to think that biofilms is a place where microbes communicate a lot between them. So the horizontal chain transfer are being magnified. So of course, we don't know the size of this. For example, in FAO, the other week we were talking to try to understand if we already got data estimation about this. Probably it's too early, but the scientific evidence, I mean, to see if there is, how much there is an effect of microplastic on the EMR issue. But for sure, the laboratory, the scientific data are showing that plastisphere is a place where the antimicrobial resistant transmission is increased. And all of this data, again, is not a single study, is a review, is showing this. And this is the kind of, from another paper, the conceptual model. Okay, so you got the soil, you got the microplastic, and as you can see here, you got the formation of biofilm, you got the interaction and the horizontal gene transfer. So the concern is that indeed there is an increase in the transmission of these antibiotic resistant genes in the soil. And again, I stress out the fact that if you got sources of antibiotics, source of manure, source of plastic altogether, this could be an issue. Also because this is a data I didn't mention before, but indeed we know quite well that for the agricultural soil, the organic biomasses, if not properly treated, can be a source of microplastic. In MINAGRIS, we are also tackling this issue. So this is an experiment that's being carried out with the group of Dimitrios Karpoudias from Thessaly, who is scientifically coordinating activities in MINAGRIS. And here we made an experiment where we look at several things altogether. We tested a plastic, LDP, and also a bioplastic, PBAT. We included also treatments with fungicide and with pesticide and antibiotics, tetracycline and sulfometazole. And we assessed, as you can see here, in time, what is happening. Again, this is unpublished data, but already presented at conference. So there were a number along 150 days, the interaction between the system were studied. And actually, as you can see, the analysis involved the pollutants degradation, the shotgun metagenomics, so again, sequencing everything, and the qPCR quantification of pesticide-degraded genes, but especially antibiotic-resistant genes. So this is some of the outcome we got. Blue is the plus, sorry, yes, this kind of blue is the plastisphere. The one in orange is in the soil. You see TET2 on the plastic and on the bioplastic. In both cases, there is a significant increase of the quantity of antibiotic resistant genes. And what we also found is that this is also happening to other genes. Probably we also see an effect of the pesticide. So again, our system is a little bit more complex because we got the plastic, we got the pesticide, we got the antibiotic resistant genes, and it's possible, as shown this data, there is an increase due to the presence of pesticides. So again, the system, of course, is complex as showing this data. Then I'm moving to the second and final part almost of the presentation. As microbiologists, we are quite interested in looking at the microbiomes, especially also in animals up to the humans, okay? And one of the questions we've been asking the last years, of course, not only us, because there is a huge amount of work, is how plastic, assuming that plastics, and especially micro nanoplastic, are being ingested, what is the impact on the organisms that are ingesting? I'm starting here with earthworms because we know the importance that the earthworms have for soil quality. And this study carried out with colleagues, Professor Mohamed Bani and other groups from Sousse University in Tunisia, is showing an experiment where we tested microplastic, 2,4-D, which is a very common herbicide, and the combination of the two in the microbiome of earthworms. So these experiments were conducted in microcos where we got the soil, the earthworms dwelling on the soil, and these compounds. And as you can see from the model, of course, there is an ingestion of micro nanoplastic and potential effects. So the data you see here is that there is a clear distinction between the gut of the control earthworms and the gut of the earthworms that were exposed to 2,4-D microplastic on both of them. Together also with other colleagues, Professor Lucini from my department, who is doing metabolomics work, we also measure a shift in the metabolome. So the microbiomes in animals that are important for soil quality are for sure changing and reacting to the plastic presence. I also have to state for clearness and for correctness that as microbiologists, we must consider that a change in the microbiome is always happening. The point is try to understand, if possible, if this change is positive or negative for the organism. A similar study with the same group in Tunisia was carried out, in this case, in marine environment, and we are looking at mussels, okay? So this is an area in Tunisia where different kind of plastics were sampled. You can see the distinction between size and different kind of plastics. So these are environmental plastics, what you find in the real world. These are pictures at SAM that we carried out, and as you can see, there are all these kind of shapes of biofilm of plastisphere on this kind of plastic. And then, of course, the effect on the gut. Okay, here there are quite a lot of data involved, but again, what we can see is a shift. And in this shift, this is the number of species that we are measuring, that we can measure through the classical NGS methods. And I have to say that, for example, in these cases, on a toxicological point of view, there were found some toxic effects. And we also found change in the microbiome, for example, in some cases, a reduction of beneficial microbes. So there is indeed an issue where we are finding that some of the effect on the gut microbiome is indeed related to a reduction. We are also carrying out study on humans. This is another study that is unpublished. We are now in the final elaboration. Well, the data are elaborated. We are finalizing it. And here together with different colleagues, University of Florence, University of Catania, and so on, we were studying again environmental microplastic, and we simulated with a system called InfoGest the effect on the gut microbiome. And in this case, we were comparing gut microbiome of healthy individual with gut individual with ACL, so sclerosis, lateral sclerosis, which is quite a strong disease, unfortunately. And as you can see, we got in both healthy individual and the patient, no plastic control, plastic coming from tap water. So this was isolated, extracted, purified from tap water, and pure polyethylene microplastic beads, okay? Number of evidence here is that, first of all, you can see on the left side are the healthy individual, on the other one, the plastic. So the sclerosis patient, and there is a complete difference between the two. And unfortunately, as you can see, the data on the right, which is the alpha diversity, let's say the total richness of bacteria, above we got the healthy and below we got the patient. The scale, of course, is the same. You can see how much microbial diversity is reduced in this individual. And what we can see is that apart from that, also in both healthy and other individuals and patient individuals, the plastic are creating a decrease in diversity. Okay, so both of them are affected. And of course, in the case of patient, the idea here is that this further reduction can be, of course, a further problem. This kind of data, since we do whole metagenome analysis of all of the genes, we also made analysis with a collaborator of mine, Filippo Vaccari, postdoc, assessment of the potential plastic degradation genes. So, for example, we use this plastic database, and as you can see, in both cases, we can see that as compared to the control, there is an increase in potential plastic degradation genes. So the point here, as I was also trying to mention with the soil, so in this kind of One Health approach, let's say, that is getting popular but important, of course, there are similarities to what is happening in the soil and also in the human gut. This means that the microbial communities are somehow adapting. There is, of course, an impact, which is negative for many aspects, but potentially there is an ability to increase the ability to withstand. And so this is, I think, is the final slide. This is just a recent paper I found from people working mainly with human gut microbiome. And now there is a kind of hypothesis, actually there are some evidence about it, that beneficial microbes, in this case for humans, but the same could apply also to soil, to animals, to livestock and other environments, can be alleviating some of the toxic effects. Also because we know that most of the toxic effect of plastic is due to inflammation and to effect of the microbiome. Some microorganisms, including the probiotics, can have some health claims. So it's getting a little bit popular, the idea that eventually with the diets, with the probiotics, it's possible to alleviate these effects. Of course, I would take these things with extreme caution, also because some of these claims are already being taken, let's say, a kind of commercial level. And I think, of course, it's not a good thing if there is to make a business. I would say if there is not enough evidence. But the message here could be that, this plastisphere, so this microbe interacting with the plastic or living the plastic, can potentially be a source of the solution. They can be a problem if we're talking, for example, about AMR, but it can be a solution because they are learning how to cope with them. For example, there is another paper here. We are talking about PFAS, not plastic, but is of course a very related issue. There is a strong evidence that some bacteria are able to absorb the PFAS and discard them with the feces and so on. If we consider that, like, estimation indicate a 1-2% or even 3% of ingested plastic that remain in our bodies and in the gut, if microbes can help to discard them, to get rid of some of them, of course, this can be a possible good outcome. So I don't know how much I talked, sincerely. I hope it was the correct length, and I would go to conclusions, okay? So first very important point is that people, scientists were thinking until a few years ago that plastic, okay, is not a problem, it's a material, it's inert, we use it, so no issue related to plastic. There are still some people that claim this, but they don't have any kind of scientific evidence to claim this, because science and evidence are showing that plastics, especially when they become smaller, when they fragment, they are biologically active, okay? So we don't have only the problem, let's say, of fish or animals getting, let's say, suffocated and so on, which is of course already an issue, but the problem is that plastic become micro and nano, both plastic and bioplastic. Is important to state this, and as smaller they get, the more biologically active they're becoming. There is, of course, an interaction. I mean, as a microbiologist, I'm interested in working on microplastic because they are both micro. I mean, both micro in the sense that they share the same size, so they are the first one interacting. And for this reason, the so-called plastisphere, including the soil plastisphere, is of interest because it can be a source of beneficial interest in microbes, for example, for future degradation, and of course to see the toxic and ecotoxicological aspects. My vision is also, I would say, as a biologist, as a microbiologist, that the microbiomes are not something that is frozen and constant. Microbiomes are constantly evolving. I always repeat that if we think about the term xenobiotic, so plastic is a xenobiotic because it's something alien to life, okay? Um, we may think that, I don't know, when milk appeared because of mammals was even a xenobiotics. Of course, much simpler, much easier to degrade. But the point is that new chemicals can arrive because they are man-made or because of evolution, and the microbes are starting to interact. So taking the example of the PATase, so the enzyme that was discovered by Japanese scientists almost 10 years ago, so an enzyme able to degrade PAT, it's possible that some enzymes are being evolving or can be discovered. For example, I didn't have time, actually, I didn't include this slide, but there is a very interesting paper on Science of 1 month ago where people used artificial intelligence. They were testing polyurethanase, so the enzyme to degrade polyurethane. And what they found, and this is a hint of how artificial intelligence can be useful, using models, 3D models and so on, they identify enzymes that were 10 times faster in degradation, okay? So this can be interesting, and as I said, metabolic pathways can be found. There is all the issue of bioplastic, but I didn't talk much about it. There is indeed a resemblance between rhizosphere animal and human gut microbiome, even if those are completely different systems. So the main point is that there are different reactions of the plastisphere in this environment, but they are very, very different environment. Sometimes people making a lot of connection claims between the connection between soil, plant, and human gut microbiome, but it's largely an incorrect claim because the microbiomes are extremely different. The point is that each environment with its own microbiome can learn to cope with some pollutants, some issue with potentially similar processes. So it's interesting and intriguing to study that together. And then, of course, it's needed to say we need the strong interdisciplinary networks. MINAGRIS is an example of that because, of course, it's focused on soil, but we got the chemists, the physicists, the communicators, the policymakers, and so on. And I believe we are in a stage where we need policy take up to tackle the issue of plastic. Reduction of its use it only when it's essential and cannot be substituted with other materials, and probably trying to find solution for solving some of the problems we are seeing. I finished with some announcement because in Minagris we are in the stage of organizing our final event. Will be a public event in Brussels. The date is September the 16th, so I'd like everybody to mark it. We are probably coupling this event with also a kind of artistic event because in Milan last year we organized exhibition. Università Cattolica organized an exhibition with Fabrizio Ferri, which is quite a famous photographer, and we got picture of Susan Sarandon, Isabella Rossellini, and so on, all of them with plastic. So hopefully we're gonna bring this exhibition together with Minagris. And as some of you, I think, know, in Piacenza, we started 3 years ago AgriFood Plus, so a conference totally devoted to plastics in the agri-food chains. We organized the second edition this year in Brussels, thanks to Luca Nizzetto, and it was quite a success because the number of people were increasing. And now I just announced it. We are defining the date yet, but we will make for sure the third edition in Uppsala in 2027. So we want to make it every 2 years. I finish with the acknowledgements, starting from my group, okay, people working or that were working in my lab. Some of the people I mentioned here, I think I forgot some, so I'm sorry about it. But as you can see, from Turin University, Turin Polytechnic, Tessali in Greece, of course, Wageningen with Violet and Esperanza that are coordinating Minagris, and a number of projects starting from Minagris, but of course, other projects and activities that can support our research. Okay, so I think that's it from me.
That was fantastic. Thank you so much, Eduardo. It's been an amazing tour of the importance of a plastisphere and how microbial activity is a really key part of this. So really important talk. Thank you. So we'll move into a short Q&A session. So first of all, we have a an anonymous question from someone
Hmm?
who says they had a bad connection, so they may not have caught everything. But they said,
Hmm.
was it that the bacteria was found in the plastic and that was degrading in the field? So was the bacteria found in the plastic and that's what was
Uh,
degrading?
Well, we didn't find it directly. The point was that we compared these different plastics. So we did the microbiome. The colleagues, in this case, Professor Frake at Polytechnic University, did the degradation, and there was the evidence that the mostly degraded plastic had a different microbiome. Okay, so the hypothesis is that this was due to the microbiomes. We also did some enrichment, and actually, the second study presented, the one on fungi, was deriving from that. So the answer could be yes. They were, again, this is a partial degradation. We're talking about probably 5-10%. If you look at the literature on polyethylene and so on, it's strongly also depending on the kind of plastic because it can be small molecular weight, low density, high density. So it's huge amount of plastic. Of course, if it's a smaller plastic in terms of density and in terms of molecular size, you degrade a little bit more, but not so much. So the answer would be yes, but we don't know exactly which microbes, and also very, very important, a very partial degradation. Okay, so this was not like a big discovery. We found a very good micro, whatever.
Yeah, excellent. Yeah, sounds like there's still more to explore there. Excellent.
Hmm.
So, Andrea Cavina has asked, what prompted the shift in perspective regarding plastics? So going from as inert
Hmm.
materials, what led to the exploration of her various interactions?
Yeah, well, once again, I would say this was mostly thanks to Professor Thompson. Actually, he was with us in Rome. We did also a winter school together with him, was teaching because once he was realizing these small particles that started the, let's say, the line of research. And the evolution of the line of research was using different kind of model system and also real system that these are impacting. Okay, so for example, in Minagris, in Papillon, we got total evidence that many functions of the soil, physical, chemical, and microbiological properties are affected. The same is happening in human. Of course, I'm not from a I'm not a doctor, right, in medicine, so I just talk a little bit about this concerning the microbiome. There are a number of evidence that are showing that also in our bodies there are some effects. What I would say is that we are not yet able to make a risk assessment. So we cannot say, okay, some diseases are due to that or not. In the model system, we show that this is happening.
Yeah, excellent. And yeah, it's brilliant. I mean, Richard Thompson, as you say, is a really pivotal person in all of this. And, we're very pleased to say that he's confirmed he's joining us at our final event in Brussels
You do?
in
Yeah, I
September.
didn't announce it. That's great.
So that will be fantastic. Yeah. So
Yeah,
hopefully we will see him there and hopefully hear from him too. So that's
yeah,
fantastic.
yeah.
So another sort of question from Cicely Barnes. So she's working in Norway, and she said among organic farmers, they found a practice of using probiotics, and they're being used to increase soil
Hmm.
microbe activity, and thus biodegradation of cover crops is increased. She wonders whether this has also impacted the degradation of the PBAT film used.
Okay.
Um, and yeah, I wonder if you could just kind of explore whether you know about this practice of using
Hmm.
probiotics on soils from
Yeah,
elsewhere, any reflections on possible effects on PBAT degradation.
yeah, I like very much this question because actually it's connected my two main line of research, let's say. One is on xenobiotic plastic and so on. The other is on beneficial microbes for
Mm.
agriculture. So in my group, we also have a spin-off. We develop biostimulants, microbes for agriculture. So it's a very interesting aspect. Actually, the two of them were not so much linked until now. I mean, boosting of microbial activity, biostimulation, and so on, and biodegradation of bioplastic. But this is an idea that we are also working on because I believe there is a potential to couple them. If you consider that they are bioplastic being used as mulching, but also potentially for encapsulation of fertilizer and so on, to use both of them together would be interesting. Also because what I can say about bioplastic from our experience and our scientific papers is that unfortunately in many cases they do not degrade enough faster. So
Mm.
anything that could be done to make them more biodegradable, I think it's more than welcome. of course, here is the problem. Also, if you speak with the people working of them, is that ideally you would need something that is stable for a certain period of time because you need that use in agriculture and after needs to degrade. So probably they are line spaces for research to trying to trigger at a certain point this process. But the question I think is really good because to couple both approaches of using microbes for the plants and for biodegradation is very interesting. Yes.
Yeah, excellent. Thank you. Brilliant answer. And yeah, lovely to hear that you do some similar work. So perhaps
Yeah,
you
yeah,
may
we can
link
also
up
get in
together,
touch
which would be
then.
good.
Also, because everybody, please
Yeah.
write me or whatever. I mean, many are also here are friends and so on, but
Yes. Yeah. Yeah. I recognise lots of names in the audience, which is lovely to see. And also great to welcome new people too. So
yeah.
Jim Harris has said, is there any evidence that microplastics are being washed down the soil profile to bedrock, potentially capping it? So he says he's thinking of applied slow release
Mm.
fertilizers and sewage sludge here.
Not well. The first part of the answer, yes, because they know to be moved. Again, I didn't mention all of it because we are doing a lot of things in Minagris, but some of the work also done with Wageningen and so on is about the movement of the microplastics. So the microplastic, they move quite a lot. Concern in the capping, I don't know because I'm not a pedologist or geologist, so I cannot answer that. I believe you would need quite a lot of amount to obtain this effect, but again, I'm not sure about the possible answer.
Yeah, yeah, absolutely. We also have a question about additives and what the role of additives are in how bacterial communities are changing.
Absolutely. We don't know yet because now the next open questions, I didn't mention it enough, is indeed the additives. So we don't know how much of the degradation, but also how much of the toxicity is due to the additives. And I think this is an important open questions also at policy level. I mean, I know many people that were that are part of the independent scientist coalition for the plastic treaty. You know, they were in Geneva this August. Unfortunately, the negotiation failed. Okay, so many people want a negotiation. They were, of course, lobbying political and economic interests, but it struck me that the two reasons that I read that there was no treaty was, first of all, reduction in the production of plastic. So some groups, some countries did not want it, and complete, let's say, information about the additives. Okay, so the scientists wanted, okay, we want the data about the additives, and unfortunately, this cannot be done because, again, it could be that many of the toxic effects are not maybe due to the polymer itself, but all the additives. And we're talking about thousands of molecules, and some of them are not known. We are also learning how to quantify them because it's complicated from a chemical point of view.
Yeah, yeah, absolutely. And on the the failure of the plastic treaty that
Yeah.
took place recently, MINAGRIS did put out a news item about this, which I've just posted in the chat. So hopefully that provides further context about what happened there. So Linda Signorini has said she's very interested in the interaction between microplastics and heavy metals in soils and how microplastics may transport those metals and increase their bioavailability. She wondered if you have any thoughts or observations
Okay.
on that.
Yeah, I know a little bit more about organic pollutants because I'm also working on bioavailability organic pollutants, and also in Minagris we are testing interaction with the organics. So the point is that plastic are mainly hydrophobic. So most of the interaction is happening with hydrophobic organic pollutants. I guess that metals are interacting as well. The main point is that I didn't mention it clearly, but scientists talk as a kind of Trojan effect. What does it mean? Micro and nanoplastic, they travel through, okay? They go into the plants, they go into our bodies, they go inside the cells. And the Trojan effect is indeed the fact that they can bring together with them both pollutants, including potential heavy metals, even if I don't have studies on that directly, and pathogenic bacteria, antibiotic resistance bacteria. So it's the whole system altogether that is involved.
Yeah, yeah, no, absolutely. That's brilliant. Thank you so much, Eduardo. And I
Sure.
think there's a lot of really exciting research that's happened here and also a lot of future direction. So there is more research needed even after Minagris. So and all the other work going on by Richard Thompson and our sister project that has now ended Papian. So I'd like to start closing up this webinar. So thank you so much to everyone who has joined. Once again, I will quickly share the image for our final event. Hopefully you can all see that. Please do save the date, 16th of September 2026. Don't risk missing out on this because we have limited in-person places. So do get it into your diaries. Registration will be opening sometime in the new year. So do sign up to our newsletter to be the first to know when registration is opened. We look forward to seeing you there. We are also planning to run a further series of webinars. So Eduardo's is the first of many. So do stay tuned. We will be announcing further webinars very soon as we pull together all of our key messages from the project. So thank you very much, everybody, for joining. Please do stay in touch. Reach out to Eduardo with any further questions. And we are also here as the dissemination and comms team for any MINAGRIS-wide queries. Okay, excellent. Okay, thank you, everybody. We finished 10 minutes early, so we can all go for lunch, which is lovely.
Okay, yeah.
Excellent.